And welcome. Good morning, everyone. So I want to talk today about biotic interactions of Baltic Sea plankton communities identified by DNA metabarcoding and network analysis. So coastal seas like the Baltic seas, they perform key ecosystem services that we humans strongly rely on. And these ecosystem services, they are largely supported by phytoplankton blooms. For the Baltic Sea, the summer phytoplankton blooms, they are usually well known. And here you see a satellite image from the central Baltic Sea, seeing these large, extensive blooms here in bluish green that expand over very large areas. And these summer blooms, they consist of filamentous algae or phytoplankton species. And these can be as big as seen by the naked eye. And these larger filamentous cyanobacteria, they are thought to be unpalatable for the zooplankton communities. During the same time, also over the summer, we also have the smallest of the phytoplankton species in the water when they reach also the highest numbers. And these are the picocyanobacteria, as you see in this slide here. And they are as small as 2 micrometers. So, and at the same time also, when we have these blooms of cyanobacteria, we also see the highest numbers of crustacean zooplankton in the water. And the most abundant of these ones are the copepod species. So what gets, however, usually less attention in the Baltic Sea are the spring blooms. Also, the spring blooms, they are of equal ecological importance, and they reach even higher biomass. And here in this satellite image that was taken in March, you see that the spring blooms, they start to develop in the coastal areas, and then they spread out to the open waters. And these spring blooms, they start as early as March, so when the water is still cold, and while there is still snow on land. But the spring blooms, they consist of very different species. So the species, they are, in this case, they are smaller size species, and they consist mainly of diatoms and dinoflagellate species. And because the water is cold, about 5 degrees or so, so what dominates from the zooplankton species are mainly protists and rotifers. These are smaller celled zooplankton organisms, as you see here, like some rotifer species, and here we have even smaller rotifer species. And they have a relatively short development time, so they can grow quicker when it's cold. So, and the phytoplankton blooms, they take up carbon from the atmosphere, and as such they regulate climate at a global scale, but also in addition they sustain the production of benthic life, as we see here from blue mussels, but also fisheries production. But the magnitude of this aquatic production, that depends largely on how the carbon is transported in the food web. And here, zooplankton organisms, they perform a key role. Because zooplankton, they graze on phytoplankton, and as such, they take up phytoplankton. They control also the phytoplankton blooms, but they take up the carbon that is fixed by phytoplankton. They concentrate it. They concentrate also essential nutrients produced by phytoplankton. And then they are themselves food for fish, and therefore contribute to the production of fisheries. At the same time, the zooplankton, they also contribute to the carbon export to the seafloor, and therefore to the carbon burial in the sediment. So, but if we want to manage the phytoplankton blooms as well as fisheries production, we need to know all this about the interactions between the plankton communities, like who is feeding on whom. But this has been usually a challenge so far. And the key question still is, who interacts with whom, or which predator is eating which prey. And for plankton communities, this has been a challenge, largely because they are very diverse. As you see here on this microscopic picture, they come in very different sizes and different shapes, and they are small. So that also means they are very but they are hard to study from field collected samples. And also in addition, they are or some cells are very fragile. And that usually means that what gets attention for the plankton communities are usually the larger organisms that have also exoskeleton and that remain as crustaceans or copepod species as we see here. So what we have established a method is that determines and allows to determine feed-on interactions within the plankton community for the first time from field collected samples. So, and how does this work? So what we do is we collect samples from the field, both of plankton and also of the water, and then in the laboratory we select individual zooplankton organisms, and also we used advanced microscopy that allows us to sort out small cells, protists, for example. And then from these individual zooplankton species, we extract the DNA that is in their gut. And then we use marker genes that allow us to identify what they have been feeding, and then we get the different taxonomic assignments of their diet. So, and this is called the metabarcoding approach, and this metabarcoding approach that gives us a very detailed understanding of the individual species interactions. And then we combine this information with the biomass data from the monitoring of the plankton communities done by microscopy that calculates also biomass. And then combining this information, we use bioenergetic network models that allows us then to estimate the fluxes from primary producers to upper trophic levels. And here, as you see in this animation here, this is a dynamic model, so that actually allows us to identify the fluxes over the annual cycle for different seasons and also for the different species. Like here we have the primary producers, phytoplankton, here we have different zooplankton, and then up to fish. So what do we find? So first of all, what we see is we have niche partitioning among zooplankton organisms. And let me first orient you in this slide. So here on the top, we see the dominant zooplankton organisms in the Baltic Sea, and they are arranged by size. So on the left-hand side, we see the larger copepod species, then we have the two dominant cladoceran species, and then later on we have the smaller sized rotifer species that reach also very high numbers. And then in the bottom, we see the dominant phytoplankton taxonomic groups, and they are also arranged by size. So they come from the smallest Picocyanobacteria to the largest filamentous cyanobacteria. And the width of this bar on the top, that tells you the proportion of the selectivity of the organisms. So what do we see? So if you look at the largest copepod species, we actually see that the substantial part is made out from the smallest of the phytoplankton species, namely the Picocyanobacteria. And also more advanced analysis tells us that that they are mainly taken up indirectly via feeding on primary consumers. But then, on the other spectrum, what we see is that for the smallest zooplankton organisms, they take up quite a large proportion of filamentous cyanobacteria. And this is really something that was very unexpected, because those cyanobacteria, they are usually thought to be unpalatable and therefore not grazed by the zooplankton community. But surprisingly, what we also see is that the diatoms and the dinoflagellates, so these are the phytoplankton species that dominate the spring bloom, they actually show relatively low proportions in the diet of the zooplankton, and mainly show up in acacia and to some extent bosmina and the rotifer keratella also. So these are mainly species that dominate also during spring. So when we use this information and combine this with the network model, we can then ask which phytoplankton groups support the production of the zooplankton. And here, again, we see the phytoplankton dynamic over the season. And here we see that in the spring, if you look at the shaded bar here, we see that the diatoms and the dinoflagellates bloom particularly. They are very high in the spring, and they reach higher numbers and biomass compared to the summer bloom. But what we see is when we look at what is being preyed upon from the spring bloom is that surprisingly the grazing pressure on the spring bloom, or what we call also the predation loss due to zooplankton grazing, is actually very low. And this is largely due because of the temporal mismatch of the phytoplankton and the zooplankton bloom. As I mentioned before, the phytoplankton blooms, they occur very early here in the Baltic Sea, when the top or the largest number of zooplankton are not there yet in the water. So we have minor contribution of the spring bloom to the zooplankton production, despite the high biomass. But then if you look to the summer, what do we see during the summertime? So in the summertime, we see that pico and filamentous cyanobacteria, they contribute largely to the production of the zooplankton community. And this is mainly due to the predation of the rotifer species, the smaller ones, but also some copypod species. So what this actually also shows, this gives us a mechanistic understanding also how the cyanobacteria carbon is entering into the food web. And this is actually quite contrary also to the belief that cyanobacteria are not being grazed, but they are rather thought to be deposited to the sediment and therefore contribute also to the anoxic spread in the Baltic Sea. So, but the question for us is still and has still been, what happens with the smaller sized zooplankton organisms like the rotifers, because we don't really see them also being preyed upon by the more predatory zooplankton organisms. So this let us then ask about, what about the fish communities? What are the dominant prey of the plankton fish communities? And here we also use DNA metabarcoding. And this has an advantage also for the planktivorous fish, because so far the fish communities has mainly been, or the predation of fish has mainly been identified microscopically. And by microscopy, we use those fragile, smaller organisms because they burst very easily and they get also digested very easily. So here we have a limited sample size of the dominant planktivorous fish in the Baltic Sea, and we have herring, spread and stichelback. And what we see is that herring and spread, they show quite the same feeding niche. And this is something that confirms also the conventional approach. What is interesting actually also that they feed also on the comb jellyfish, Mertensia. So this is actually also a link that has not really been identified before. But what is interesting is that the stichelback, a species that has exploded over the last decade in the Baltic Sea, it seems like it has its own feeding niche. And this is a species that utilizes like mainly rotifers and also some other type of copypods that other fish species don't really use. So this actually means that this species occupies this same niche, and that may also contribute to the success of the species. So how does this contribute to ecosystem management? So what we see is that the spring blooms, they support only low production of the zooplankton. So this actually suggests also that the spring bloom, they contribute largely to organic export. And this is also something that has been confirmed by sediment traps, that they find the largest input during or after the spring bloom. But then what we see is also that the small-sized zooplankton organisms, mainly rotifers and ciliates, they are an important link between phytoplankton and fish at a very crucial but limited time of the year, namely in spring, when we have also fish larvae in the water. However, they have very low grazing pressure, and therefore they have quite a minimal regulating impact on the phytoplankton bloom. What we also see is that the copypod zooplankton species, mainly Temora and Acacia, they have quite a high regulatory impact on the summer cyanobacteria bloom, and therefore to a certain extent also control the summer cyanobacteria. And overall, the studies also suggest that any measures to increase the biomass of zooplankton during the springtime will reallocate the energy to fish, because during the springtime we have the small-sized phytoplankton that are very energetic and efficient food webs to higher production. So with this, I want to acknowledge my PhD student that performed this work, and also the funding agency. And if we have time, I'm happy to take any questions. And thank you for your attention.
Thank you, Monika. We have time for 2 questions. Yes, Francisco.
Yes, thank you, Monika. I was again, as you mentioned, surprised to see that the smallest zooplankton eat the filamentous cyanobacteria. So physiologically, how does this happen? Or do you think that this filamentous cyanobacteria needs to be broken down before the small zooplankton
Yes,
can actually
right. So this is a good point. To a certain extent, we assume that they graze on the degraded cyanobacteria, also the filamentous cyanobacteria, to the broken down, because we have also some indication that the detrital food web is quite strong in the Baltic Sea also. So this is also something very special here in the Baltic Sea that we don't see, for example, when we look at the west coast. But then there is, to a certain extent, also direct grazing. And I think this has been shown from laboratory experiment, actually, for some rotifer species, but also for some ciliates. So they are able to take up these filamentous cells, like spaghetti eating, and they just incorporate them and roll them up actually in the gut. So I have a colleague of mine, he investigated this once in the lab, and they just have been really sucking them in like spaghetti eating, so to say. So that is physiologically, it is possible. Yeah.
Stop. Have you studied the nutrient value of if it changes in acacia, for instance, when they are grazing cyanobacteria instead of other phytoplankton?
This is a very good question, actually, also the nutrient value, because that comes in another aspect. Is cyanobacteria are known to be of low nutritional value because they lack essential nutrients. They lack essential fatty acids that are very important for fish production, and also for us humans. So so I think what we learn is actually that the transfer of these essential compounds up the food web is actually a little bit more complex. And sometimes, so there are definitely also other phytoplankton that contribute to the quality. But then also the cyanobacteria, they are very good carbon source. Yeah. So and what we see sometimes is that if the zooplankton, if the majority is cyanobacteria, but if they get a minor proportion of other phytoplankton that has these essential compounds, that they are actually maintaining also the production. Yeah, but the question still is, and I think this has not really been identified for the Baltic Sea, is what is the nutritional value of the individual zooplankton organisms? And we have studied this actually for benthic organisms currently, and what we see surprisingly is that the fatty acid composition of benthic organisms is not determined by phytoplankton input, as it has been always believed. But there's a very strong intraspecific difference actually also. So that means that the organisms, they may have also some effects actually to regulate the essential compounds. So I think it's a little bit more complex than just this straight line actually also, that there is quite a lot of individual mechanisms also for the secondary producers.
And we also have a final question from the Slido app. Where are the cladocerans in the fish diet? Based on stomach analysis, cladocerans are important diet for stickleback.
This is actually quite a very good question, the cladocerans. And here I would actually need to go back to the slides. And what we see is they have not really been here, not in this time. But I should actually say that this data set comes from a very limited number of samples. And this data set comes in spring, and the cladocerans, they usually peak during the summertime. So we have now a new project that we just start now this year, where we actually look into this interaction between zooplankton and the planktivorous fish in more detail, and we also investigate individual life stages of the fish. So the reason they are not here is because they simply were not in the water. We were too early here. This was done in April or so, when the cladocerans are not there. And usually, I should also say, there's lots to say about plankton, is that the cladoceran peak is usually very short. Yeah, so you really need to be there at that time the year if you want to get them. But they are most likely important.
Thank you so much.